started for our next talk. I mean, as usual, it's end of the day, second day, and we have people dropping off. I did this too. I'm sorry, I was guilty of the same. You know, I had to pick up my daughter sometimes from school or daycare. I just snuck out and left the, left the show. But that's what we have. You know, we are getting to the end of the show. There is a little bit of attrition there that is going on. But I want to promote again the actual continuous manufacturing that is happening with tableting that we have as a demo. We had 8 people signed up. Now 3 dropped off. It's down to 5. We need more volunteers to get it at least to 10. If you want to see, you know, what Richard said earlier about continuous manufacturing, this unit is actually continuous manufacturing with direct compression. So it is using the same extruder, as a matter of fact, and feeders into that extruder that further brings the material into the tablet press, all in one unit. But on top of it, it has inline detectors for content uniformity. Basically, real-time release can happen. So I'm promoting a little bit that event. If you are still interested to go there, you know, sometimes you can beat the traffic, in my view. You know, not going too early, you are going to be stuck in the traffic. Anyway, so that event is still ongoing. You know, we are trying to see if we can get it to 10. So if you want to attend, please let me know. So our next speaker, my apologies again, Dr. Gu, I wanted just to make sure that I have that announcement. So our next speaker, which brings us to one before last presentation, is going to talk about bioavailability again, but about the protein degraders delivery. Dr. Gu currently is working at LifeMine Therapeutics as the head of CMC and quality assurance. He has an extensive background in pharmaceutical industry. Prior to joining LifeMine Therapeutics, Chong-Hui held roles at Foghorn Therapeutics, Aegis Pharmaceuticals as head of CMC, and program leader at, and also at Vertex Pharmaceuticals as a research fellow. Dr. Gu also gained experience at Bristol, you know, not far from here. Yes, were you here? You know, BMS right around the corner here, as a senior research investigator. He has educational background that includes attending the University of Minnesota from '96 to 2001. Again, thank you so much, Dr. Gu, and I leave the stage and I let you to get on the floor. Okay. Thank you. I appreciate it. So, I shake your hand.
Sure. Again, thank you for staying for last part of the conference. So I hope my presentation will be useful. So feel free to raise your hand to ask questions during the talk. So currently I work for LifeMine, but the work is mainly done at the Foghorn, so that's why I still use my Foghorn template for the presentation. So PROTAC, also known as a protein degraders, is a new modality. It's a small molecule, but it contains 3 parts. So it has a part that binds to the E3 ligase, and the part bind to the target, and then you need a linker to link these 2 binders. So the molecular weight of those molecules is about 1,000, plus minus. And it has kind of unique properties. It is more selective. It can degrade the protein instead of inhibit the protein. So that open the door to some kind of non-tractable disease proteins. And also potentially, because you can design it, so right, you need a binder, you need and a linker, so it's more specific. So it will be faster than molecular glue approach, which you basically just screen the compounds. But the one challenge for the protein degrader is the oral bioavailability, because they're big, and also it's actually lots of polar surface areas. So delivery becomes challenging. So in this talk, I will share some of the approach that we use to deliver protein degraders. So first, let's start with oral absorption. And here I kind of lay out the barrier for oral absorption. So first, the drug has to dissolve. So that's really the solubility and dissolution problem, which formulation usually can play a role to help. And then the drug has to permeate through the membrane, epithelial membrane. So that's the permeation issue. And then they also need to survive the gut metabolism, and then the first pass metabolism by the liver. So in order to deliver a drug, it has to go through all these barriers. And that will be important actually to understand what's the rate-limiting barrier for your compound, and then you design the solution to overcome that. So here's basically the approach we can use to deliver poorly oral bioavailable compounds. So I just realized I made these slides 19 years ago when I joined the Vertex. And somehow the technology I listed here, we are still using them. So it's kind of, to some degree unfortunate that we haven't really invented new ways to deliver compounds. I mean, we made lots of progress in technology, like continuous manufacturing, but in terms of the delivering compound, we haven't really made lots of progress. So as you can see here, lots of time actually, formulation cannot help, right? Let's say if it's a gut metabolism, it's a first pass, the chemist has to make a better compound. And the area formulation can help is really in the solubility region. And so, and you can use exotic formulation. At those times we call it exotic, but this time becomes more regularly used, like amorphous, lipid, nano, things like that. Or you can make a salt or co-crystals to make a material more soluble. So based on my experience, again, also based on the kind of the approved drugs, the approach people have used the most is really salt and co-crystal and amorphous to overcome the solubility issue. And we actually, back again at our Vertex days, we usually have a compound and then we will just screen all different formulations, like a nano, lipid, amorphous. And if they can make salt, we'll make salt. And almost all cases, the amorphous actually performs the best. Almost all cases. So these days, amorphous really becomes the most widely used technology to overcome the solubility limit. So this is the system we developed at Vertex. Actually, it's basically how to screening excipients. I think these days, actually, a few CROs, CDMOs, they can do that also. So basically, the idea that you can quickly dissolve API and the polymer together, and then you do a fast solvent removal so that you can do it in the 96 well plate. And then you can add the solution, you look at the dissolution of the amorphous dispersion, see how fast they can dissolve, and then you can also put on stability and run the XRD to see if the material is stable. So you don't need a lot of material to do that. And this is, again, the two-stage dissolution to mimic how the formation will go through the GI system. So first, you use simulated gastric fluid, and then you add a simulated intestinal fluid. Again, many CDMOs now can do that for you if you ask them to do it. So again, based on that, you basically look at what's the concentration over time, and then usually the faster the dissolution, the higher the concentration, the better. So that helps you pick the formulation that you want. And we can also use GastroPlus. Again, now becomes quite routinely used, that you can model basically how the drug dissolving in the GI and get absorbed, and link that to the PK profile you have. So again, bigger companies, they usually have a biopharmaceutical group can work with you to help you understand the PK profile from dissolution point of view. And if you work a small company, you can work with Simulation Plus. They have a consulting arm that can work with you to build those models. Again, this can help you understand how's your formulation perform and the difference in the PK, what's causing the difference. And that helps you design your formulation. So one example here, basically we have 4 different types of formulation. We have a lower compaction tablet, we have a high shear wet granulation, we have capsule. And so again, you also have a solution, which is basically immediately dissolving. And we can use the model to predict. The C factor actually is the dissolution factor. So we can use the model to predict basically how important is the dissolution on the Cmax and the Cmin. And you want to have the basically the Cmax not to be too high and the Cmin to be high enough over IC90. So that's the kind of optimum range. So that can help you design your formulation properties to hit that target. So I talked about the formulation on the solubility part, but what about other barriers? So prodrug, people use that to overcome other barriers. For example, the so-called the mimic prodrugs. So they are trying to use the lymphatic absorption pathway. There's actually a new company called the Seaport Therapeutics in Boston. They're actually using that technology, try to oral deliver difficult to be absorbed drugs. And then for permeability, as now the GLP-1 becomes very popular, Rybelsus by Novo Nordisk, actually they use the permeability enhancer snack. But actually, interestingly, the bioavailability they have is only 1%. But because the drug is very potent, so 1% actually is enough to make it a commercial product. And by the way, actually, ibrutinib, if you know that, that's one of the most prescribed drug for leukemia. It only has 3% bioavailability. So sometimes bioavailability may not be the problem, as long as your drug is potent enough. And then also recently Merck actually published, it's Merck MK0616, it's a peptide. So they use both ribosol and the sodium caprate to increase the permeability. And the drug is currently in phase 2 trials, and they show that you can increase the permeability by about 2-fold. Still low, but again, all depends on whether the compound is potent enough to make it work. And then people also use now the CYP inhibitor, or transport inhibitor. So the CYP inhibitor already used in the commercial product. So the well-known Paxlovid for COVID. So Pfizer actually used the ritonavir to inhibit the CYP inhibition, CYP metabolism, to make the drug half-life a little more manageable. The transport inhibitor, we use that in preclinical study, but so far we haven't used that in clinical studies yet. So these are basically, as I understand it, the current technology that we have to overcome oral bioavailability. But what if you try all these, still it doesn't work? You can tell the chemist to make a better compound, or let's say, let's try another way, right? So that's why we think a long-acting injectable may be an approach to do that. Because, as you know, injection product actually is not an issue for commercial. If you look at many commercially very successful products, like Keytruda and those products, they're giving IV once every 3 weeks. So as long as you can give a product less frequent, whether it's IV or sub-Q, it's actually acceptable. So we think maybe we can do that way by delivering that orally not available compound using a long-acting injectable, so you can deliver for, let's say, once a week or once every 4 weeks. And potentially, actually, even can be better because a long-acting injectable, the PK profile is very flat. So you avoid this kind of Cmax Cmin issue. So for a drug with a narrow safety window, that's actually can be an advantage. And also you can potentially improve the patient compliance because again, for daily pill, people may forget to take. And for long-acting injectable, it's less likely they forget to take the drug. So far, the long-acting injectable actually has been used in multiple disease areas. Most used still in the kind of the antipsychotic, which makes sense, right? For people have schizophrenia, you cannot ask them to take a drug every day. So the long-acting injectable kind of makes sense. And then now recently, actually, in the HIV field, as you can see in some TV ads, the long-acting injectable start to replace the daily oral because actually the HIV patients prefer to have less frequent injection, like once every month, or now Gilead has product once every 6 months, instead of taking the pill every day. And there's one kind of psychological effect, like if you have to take a pill every day, it reminds you you have a disease, but if you only need to take an injection, you can feel you are normal, just like everyone else. So all these arguments kind of support that if you can have a long-acting injectable product, it can be commercially feasible. So the technology that's available now for long-acting injectable, one way is you actually modify the API to give it a longer half-life. So people are working on antibody drug conjugate. Or antibody degrader conjugate. So once they conjugate to an antibody, it becomes antibody-like half-life. So it becomes once every 3 months type of injection. And another way is you pegylate the API. Again, that makes the half-life very long for API. And then a more traditional way is actually using sustained release formulation. So you can use suspension, usually aqueous, and lots of time they use a nano suspension. So the drug, you inject the drug and then it becomes a depot, the drug gets slowly released. And another popular way is called in situ gel. So the polymer drug dissolving in an organic solvent like NMP or DMSO, and you inject that, the polymer is not water soluble. So once you inject, they precipitate, forming a depot, and then the drug and the polymer is biodegradable, and then the drug will slowly release over time. And another traditional way is called the microsphere. So basically, biodegradable polymer, you incorporate the drug into those PLGA type polymer, and then you make a microsphere, and then you can inject the microsphere, and then the drug gets released slowly. Or you can have implants, but that's usually for kind of very long release, usually once a year type use. So I work for discovery stage company. So the question people ask, so all these commercial products, usually they have oral product first and then they convert to long-acting, which is kind of easier. But now if you want to have a new compound, especially at the discovery stage, can you use this technology to support discovery? Because you don't really have lots of compound and you need a very fast turnaround. And so we look at that. We think that there's 2 types of formulation we can use. One's in situ gel, which basically is a solution formulation. So you need a very small amount of material. You can make that. And another one is suspension, nano suspension especially. Basically, you need a nanomil. Again, these days they have very small nanomil called a delta vita 1. You just need a milligram of material. So you can make that nano suspension and then very quickly turn around to support this discovery. And so we actually, at the Foghorn, we extensively use that to support discovery. Now actually we have a drug candidate soon, probably will file IND sometime this year. And then from PK/PD perspective, sometimes you may not have the final formulation, right, early stage, so you don't know how to do the PK/PD, but you can, assuming that the long-acting injectable is more like a continuous infusion, so you can use those to model your PK/PD to understand how the drug will work for the target. So here is basically showing how the in situ depot formulation works. So basically, as I just said, you have a polymer drug dissolving organic solvents. They are not soluble in the aqueous medium. So once you inject it into either subacute or intramuscular, they precipitate, and then they forming a depot, and then the drug can get slowly released. So mostly it's using polymer precipitation. PLGA is still the most widely used polymer for this purpose. But people also start to use a lipid, like a phospholipid, as a way to generate that precipitation. And there's a less used technology like just in situ polymer cross-linking. So you will deliver actually monomer and ready and initiate together, so they will in situ forming the polymer, or using a thermally induced polymer precipitation. There's a polymer that's more soluble at a lower temperature, and it becomes a solid at 37 degrees. So you can also use that. In terms of suspension formulation, probably the first long-acting was back in 1960s, those for hormones. So people actually use oil to do that. But the oil was found to be painful. So these days it becomes a less popular approach. So people use aqueous suspension more these days. So aqueous suspension is basically you mill the material, control the particle size, and then because rely on the solubility of the compound to slowly release the compound. So Carbinuva, that's a GSK HIV product, is made that way. So you just mill the material. And the lab scale, you can do it at a milligram scale, and there's a larger scale nanomill available that you can make tons of the material. So one question people also ask, right, you have lots of choice. What will be the formulation choice? In our case, actually, we typically just try both to see which one works the best, suspension and CQ gel. But there's a review paper published by Pfizer kind of showing, just look at the commercial product, what technology people have used. They found that for poorly soluble compound, people most likely use aqueous suspension, which makes sense, right? Because the material is not soluble, so it will slowly release anyway. So you don't need actually other things to help. And especially if you have a high dose, the aqueous suspension also works better. And then for a soluble compound, they usually use the in situ gel because you need a polymer to slow the release. So in situ gel makes more sense. But in our case, actually, our compound is actually very insoluble. So sometimes the aqueous suspension doesn't work because it's not, so even as a suspension, just the release too slow. So you don't get the target profile. So in our case, we actually sometimes use in situ gel for very poorly soluble compound, which actually can promote the dissolution of the compound. Yeah, this slide is more for the PK/PD simulation. I will skip that. So, but in terms of what properties that we need to have a successful long-acting injectable, I get this question a lot from chemists. So really, we have to know what's driving the pharmacology effect, right? What's the theming we have to maintain? Because the PK profile is a flat. So you want to know what kind of the concentration profile you want to maintain. And to achieve that concentration profile, typically actually it's driven by solubility of the compound and and interstitial protein binding, clearance, and volume distribution. And actually, GastroPlus has a model that you can model that. So in the GastroPlus model software, they now have a long-acting injectable module that you can model how the long-acting injectable works. And a general kind of rule of thumb that you need a highly potent compound, because the higher the potency, the lower the concentration you need to maintain, and that makes it easier. And usually chemists are pretty good at making high potent compound, because so that becomes more like a single effort instead of they needed to look at different properties. So this is just the GastroPlus modeling that we have done for our long-acting injectables. You can either use, they have a subcutaneous module or the intramuscular module. So the way we do it is we use the IV data to set up all the PK property for the compound, and then you can simulate how the release rate affect the PK profile from long-acting injectable. So not too different to your oral modeling. So again, you also can do the parameter sensitivity analysis. So again, it's a built-in function in the GastroPlus. You can look at how will the, say, the particle size distribution or the solubility affect your release profile. So again, this gives you the idea how the PK profile will change. We also try to do the in vitro, in vivo correlation, but so far we found that it has been at least quantitatively difficult. There's a general trend that if you can have faster in vitro dissolution, you will have higher % bioavailability in a given time frame. But quantitatively, we found it's quite different. So we still quite use rat study a lot just to kind of screening formulations instead of using the in vitro study. So here's one example, FHD 609. It's a protein degrader. So as you can see here, on the left side, it's the PK. So the blue one is the IV, so typical IV, very quick drop. And then the red dots, these are the long-acting injectables at different dose. So you can see the PK profile is quite flat over time. And this is, see, 360 hours. So it's over about 2 weeks time frame. And then on the right side is actually the tumor growth inhibition. So the one that's growing is the placebo, and then we found that basically long-acting injectable at different concentration can all achieve similar target tumor inhibition, about 87%. And this is really look at the same compound. We look at the PD, pharmacodynamics, and you can focus on the right side. So basically, it's showing that at different concentration, all the protein is degraded. And this is another model, same compound, 609. We use a PDX model, and it's basically showing that at different concentrations, the long-acting injectable can achieve the same tumor growth inhibition as a twice-a-week IV dosing. And this is another compound. It's a CBP project. Again, it's for tumor, anti-cancer. So you can see that basically by giving a long-acting injectable, you get a very flat PK profile, and we can actually inhibit the PD in a sustained way. And then we don't have the tumor growth inhibition data here, but it is based on PD that we know that long-acting injectable will work for this program. And because we work on new compounds, so the challenge actually we facing is mainly on the first in human dose projection, because lots of time For commercial products, the conversion from the current product, oral, into a long-acting injectable, so that they don't really have that issue. But for our new compound, the first-in-human projection becomes difficult, especially the release profile of long-acting injectables is quite different from different animal species. So you don't know which animal species correlate to human. So our approach is basically just using either the average or a range of release profile to mimic how the long-acting injectable can work in human. And there's a published paper also. People have suggested instead of using a single Ka absorption rate constant to predict what will happen in human, you can use a Weibull function because the release profile is more complicated for long-acting injectable. But it's all doable. As actually, for my current company, LifeMine, we just moved a new compound into long-acting injectable as a first-in-human. So we just started first-in-human trials. And in terms of safety for long-acting injectable, so one is the dose dumping. That's always the concern. But since we are working on actually poorly soluble compound, so that the dose dumping risk actually quite low because the drug is so insoluble. So intrinsically, it's difficult to get the drug absorbed quickly. And another question regulators sometimes ask is how you stop dosing if you start to see side effect, right? That's especially for your first-in-human trial. So one argument, it's just like a long half-life drug, like ADC. So you have to start a low dose, slowly go up to see if your drug is safe. And another way is that for early stage clinical trial, we actually try to use a more faster release profile, formulation instead of the target. So let's say if you want once every 4 weeks, but for first in human, we probably go once every week. So in that way, we can stop dosing if we see issues. And for some in situ gel technology, you can also surgically remove the drug if needed. So yeah, so that's the end of the talk. So I think for oral, we already understand a lot, even though we still use the older technology, but it's important for us to understand what's the rate limiting for oral, and then we can apply the appropriate technology to overcome that. But if we cannot deliver it orally, I feel long-acting injectable, at least it's an alternative option for some high-value targets. Yeah, so we also, again, at both Foghorn and LifeMine, we kind of demonstrated that it's possible to use long-acting injectable as a tool to support discovery and to have a candidate directly using long-acting injectables for first-in-human. The main challenge, again, still because we are kind of new in this area, right? So the human dose projection, the safety, how to run that. So it's still a learning phase. But it's all overcomable. And finally, I want to thank my colleagues at Foghorn who did lots of work. Abila, Xiao Huang, Ashish, Brian, May, and Steve. So thank you.
Yes, Yogesh.
Thank you. Very nice presentation. You mentioned that suspension or nanosuspension can be used at a very early stage as a long-acting parenteral, but sterilization of this suspension is one of the concern. So which method of sterilization you use for the nanosuspension?
It's usually radiation. People use the radiation treatment, like a gamma beam and an E-beam.
But treatment. For aqueous media, still you can use gamma?
Mm-hmm.
There's no impact.
Yeah, that's how carbonuva, and there's a so-called aseptic nanomilling also, but it's less widely used. Mostly people just use radiation.
And one more question I have regarding the GastroPlus. the LAIs we also use for peptides. So do you think Gastro Plus can also be used to predict the absorption kinetics for peptides?
Oh yeah, they have the LAI module. They're actually quite active in that field. You can see lots of publication from them using Gastro Plus for light injectable.
Thank you.
Any additional questions for Chong-Hui before we move on? No. All right. Thank you very much, Chong-Hui.